questions at the end. So keep that in mind. Keep your questions to the end of the talks. And if we have time at the end, we can try to have a more like broad discussion with the 5 speakers. So we're going to get started.
Our first speaker will be Belén Pardi. She's now in Germany. It will be our first virtual talk. And she works at the Institute for Physiology at the University of Freiburg and also at the Max Planck Institute for Brain Research. Belén, are you there?
Yes, hello, thank you. So I will, do you see me as well? I will share my video, my presentation. Do you hear me well? Okay, I hope you hear me well. If not, you will let me know. Okay, it's a pleasure to be here, at least virtually. I would love to be there, but well, it has to be virtually this time to share my work entitled Thalamic Top-Down Control of Sensory Neocortex for Associative Memory. The question I'm interested in is in sensory perception and how it is shaped by experiences. For example, if we hear this sound, you probably perceive a duck. So how does our brain accurately perceive a duck while we're only hearing and capturing sound waves? This purely sensory information that is captured by our sensory organ, the cochlea in this example, is transmitted through the brain in a pathway that we call bottom-up until it reaches sensory cortex, in this example, auditory cortex. But to make sense of this external, purely sensory, in this case, auditory information, we need to integrate this information with information that resides inside our brain that is transmitted through a pathway that we may call top-down. It's information that relates to that external stimulus that has been acquired through previous experiences and that it relates also to our behavioral demands. So to perceive accurately, we need to integrate this bottom-up sensory information with top-down information. And this has been studied for some years now, and we believe that this integration takes place at sensory cortex. However, we have very little knowledge about how really accurate perception takes place at the level of the microcircuits. And I would argue partly because we have little knowledge about what top-down circuits mediate these functions. Therefore, in my work, I aim to find a source of memory-related information to sensory cortex. And now I'm showing you a cut of the sensory cortex where you can see again the bottom-up input coming from the first order thalamus, impinging mainly on deeper layers. And to look for a source of top-down information, I focused on layer 1. Why layer 1? Because you can see it's a unique layer within the neuropil. Within the neocortex, it's mainly comprised of neuropil. It contains a wide range of long-range afferences coming from other cortical areas, from neuromodulatory centers, which have been implicated in contextual modulation, and also afferences from the higher order sensory thalamus. And this piqued my attention at the beginning of my work, because we really know very little about higher order sensory thalamic nuclei. These are distinct nuclei different from the first order nucleus that I mentioned before. Higher order sensory thalamus projects mainly to, in a widespread function, and mainly to layer 1, as shown by this schematics from this paper. The neurons from the higher order sensory thalamus are multimodal. They respond to different sensory modalities. And it has been difficult to study them, largely because they're very silent during anesthesia, and most studies in the past in vivo were done under anesthesia. Therefore, I focused on higher order sensory thalamic projections to cortex, and in particular, in the higher order auditory thalamus, which had been implicated in associative field memory. However, the afferents to cortex had unclear function at the moment. However, to study the higher order auditory thalamus has a complexity that it is comes from the anatomical perspective, which is depicted here again in this figure from this paper. In this paper, what they did was to place a dye on top of layer 1 to label retroactively the neurons that are projecting to this layer, to highlight now this way the higher order nuclei. And as you can see, the higher order nuclei of the auditory thalamus are actually several, And I apologize, I'm not entering into the details of the names of the nuclei, but there are several, and they surround the MGV, which is the ventral division of the medial geniculate body, which is the first order auditory thalamus. So there are several nuclei surrounding the first order nucleus like a shell. So it's quite complicated to investigate them. So the first thing I did was to develop a strategy to investigate the projections of the high order auditory thalamus to auditory cortex. And as you can see, what I did was to look for a strategy to target expression to higher order nuclei. And you can see this in this picture of immunostaining of a mouse auditory thalamus. You can see the GFP is only expressed in the high order nuclei and avoided in the first order nucleus. And the way I did this was by identifying a marker for higher order neurons, and this was carotinin. So I used viral vector conditional expression of proteins in carotinin cremice, and this way I could now study specifically the projections of the axons to the cortex. That, as I have shown you before, the axons project in a widespread function, mostly to cortical layer 1. And I focused in the area of auditory cortex where the axons project most densely, which is secondary auditory cortex, and mainly to layer 1, as quantified here. So now using this tool, we asked, what is the role of the higher order auditory thalamus in a auditory associative fear conditioning? And these experiments were done by Teresa Spano, who this time expressed tetanus toxin to inhibit the synaptic output of the higher order auditory thalamic neurons. So she did auditory associative fear conditioning that most of you might know, but just briefly, it consists of a habituation phase where two sounds are presented, the CS plus and CS minus. In fear conditioning, one of the two sounds is contingently presented with a foot shock. And to measure the auditory associative memory, you put the mouse in a new context and you present the sounds again, and the mouse will freeze if it shows a memory. So this is the behavior you can see in the GFP expressing mice as a control. Here, the response to the CS plus, so high levels of freezing, of fear memory, as some degree of memory for the CS minus. However, the mice, when you inhibit the synaptic output of the high order auditory thalamus, they show reduced fear expression. And this effect is not present in the case of contextual memory recall. Therefore, the high order auditory thalamus is required for auditory associative memory, and this is actually confirming with this technique results that we already knew from other less specific previous experiments from other labs. Okay, so now we next asked the question we're mostly interested in: what information does the higher order auditory thalamus send to cortex in this process? So the best way of addressing this question is to directly look at the synapses that the auditory thalamus sends to the cortex. This time I express in GCAMP6, implanting a cranial window on top of auditory cortex and placing the mice under a 2-photon microscope to do calcium imaging. Like I'm showing you in this example, on top you can see the imaging of the thalamic boutons in the cortex, and in the bottom you can see how I monitor the eye pupil dilation as a readout of behavior. So this way we can look at long-range synaptic activity in vivo, and also we can look at long-range synaptic plasticity in vivo, but by imaging and following individual synapses, presynaptic boutons over days, and comparing the habituation session with the memory recall session. And the way I found this, the reason why I found this interesting is because you can do imaging of the boutons of the sound responses in the exact same conditions, such that the bottom-up content of information of the sounds is the same in the two sessions. But what is changing is that between the two sessions is that given that there is learning and associative learning in between, then the sounds acquire a top-down meaning. So this allows us to study in a controlled manner top-down encoding of information. So I developed a pipeline to follow individual identified boutons, like this example bouton, which in this example, it did not show any responses to the CS plus in this case in habituation. However, it showed robust responses to a CS plus in memory recall. And the same way I'm showing you this example bouton, I'm showing you here hundreds of boutons from several mice. in a paired fashion. So you can see a number of plastic changes. For instance, butons that were not responding, that started responding, and actually the number of butons was almost doubled. You can also see in the average of all the butons, these are the traces. In dark blue, you have the memory recall. You can see that there was a strong potentiation, and also the timing that changed, becoming much more time locked to the sound onset. So potentiation is quantified here. There was a very significant potentiation for the CS plus and a potentiation that was not significant for CS minus. So it was clear, but not significant. In terms of the timing, what's interesting is that while in habituation, the sound responses were mainly sound off responses. So this response started increasing mostly after the off of each sound pulse, because this was a train of sounds. I forgot to clarify that. In the recall, the responses started increasing already with the on. So the sound responses became much more time locked to the onset of the sound. And this is quantified as the decrease of the latency here. Another thing that we looked at is at the trial-to-trial noise correlation. So basically looking whether if you take any pair of butons, whether their behavior over the trials goes together, correlated or uncorrelated, if the butons increase or decrease together. And in this example pair of butons, you can see that these two butons were very well correlated in the habituation, and this correlation was lost in the memory recall. And actually, we observed this particularly, this decorrelation, we observe it particularly when we compare sound responsive butons with non-responsive butons, indicating that the sound responsive butons become decoupled from the rest of the butons in memory recall. And according to theoretical framework, this could imply that the butons are thus transmitting a larger amount of information in memory recall. So together, these plastic changes suggest that the high order thalamic input to the cortex will be affecting cortex much more strongly in a recall when the sounds have acquired behavioral relevance. But at this point, you might be wondering, well, but how does this plasticity really relate to the memory? Because some of you might be surprised by the fact that there are also some plastic changes to the CS minus. So we address this directly by looking and comparing the sound potentiation, the button sound potentiation with the memory strength. I'm showing you here again the plot I showed you before, and here I'm showing you now a plot of the freezing response in an additional memory recall session in freely behaving conditions. And maybe you can already notice that there is some degree of FIA memory also to the CS minus. And to compare more directly both measures, I established a measurement for FIA memory in head fixation, which consists of looking at the pupil dilation. I don't have time to enter into the details, but now when we quantify the memory strength that the mice display at the same time in which we are imaging the butons, we see that there is a very strong correlation, such that if there is a strong potentiation of the sound responses at the level of the butons, there's also a strong memory. And if there is no potentiation, there is no memory. And this is independent of whether the sound was a CS plus or the CS minus, indicating that the button's plasticity is predictive of memory strength. So this brings me to the main result of this part, of the first part, which is that the high order auditory thalamus transmits top-down memory-related information to secondary auditory cortex. So next, we asked, how do thalamic top-down signals interact with auditory cortex? And we have only started addressing this question by first asking, what are the synaptic targets in auditory cortex? To address this question, together with Joanna Bogenstall, we focused on layer 1 interneurons, layer 2-3 pyramidal neurons, and layer 5 pyramidal neurons. This time she expressed channelrhodopsin in the neurons and focused the light such that she could stimulate only the synapses on layer 1. And in these conditions, she found direct excitatory currents in these 3 types of neurons. Therefore, the higher order thalamic afferents can excite both inhibitory and excitatory neurons, and thus recruit both excitatory and inhibitory, as well as disinhibitory mechanisms in auditory cortex. But another way of interacting with the cortex actually comes the other way around, and it comes from the cortex. this time I was attracted by the fact that these axons span different cortical areas in a widespread function, as I mentioned before. And I wondered whether they are transmitting the same information to the different cortical layers. And I hypothesized that it could be the case that if they receive local modulation, then the information would actually be different. And I hypothesized that this local modulation could arise from layer 1 interneurons, since they are in very close proximity to the anatomical proximity to the thalamic axons. And it has been shown that at least one type of layer 1 interneurons can exert inhibition in the form of a cloud of GABA. So I hypothesized that the axons could be inhibited presynaptically through GABA B receptors. To test this hypothesis, I first did recordings in acute brain slices to test whether the axons can be inhibited through GABA B receptors and patching postsynaptic neurons. And this was indeed the case. When I placed the agonist, the pulse ratio changed, and also the amplitude decreased. This you can see in the quantification, in black the amplitude and the pulse ratio. And the changes were reversed by the antagonist. Then I tested whether this could indeed occur in vivo through layer 1 interneurons. And for this, I used a line that we characterized at the Let's Go lab, which uses neuron-derived neurotrophic factor, NDNF, as a marker for layer 1 interneurons. So I crossed the carotene in mice with NDNF fluid mice, and now I express GCMP6 as before in the thalamic axons, and also crimson in layer 1 interneurons, such that I could stimulate layer 1 interneurons and record the thalamic responses. And this showed that whenever in interleaved trials, I stimulated NDNF neurons, the Bouton's responses changed, the responses decayed, and also the latency changed. Therefore, layer 1 interneurons can exert control of the activity of the axons, and to understand how this control affects the transfer of information to the cortex, we collaborated with Laura Belén and Henning Spreckler at Technische Universität Berlin, who developed a computational model. And basically, simply what the model showed was that given an input of a certain frequency, what the postsynaptic neurons will read will depend on the levels of NDNF neurons activity. The main result is summarized here, which tells us that the stronger the NDNF neurons activity, the better the faster frequencies will be transmitted to the postsynaptic neurons. So layer 1 interneurons exert a local control of the frequency transfer. So to summarize what I've told you in this short talk is that the higher order auditory thalamus transmits top-down memory-related information to secondary auditory cortex, that it recruits layer 1 interneurons, layer 2, 3, and layer 5 pyramidal neurons, and that it can receive local control of information transfer by local cortical NDNF layer 1 interneurons. I would like to acknowledge the people that were part of this work. This work was carried out at the Let's Go lab at the Max Planck Institute for Brain Research and University of Freiburg. And I would like to acknowledge funding. And I would like to also share with you that I am starting right now my lab in Paris. So I'm recruiting, so I invite you to write me if you're interested in joining us. Come to Paris. Thank you.
Thank you very much, Belén, for your nice talk. And if there is any question in the public, in the audience, perhaps you are shy. There's no question. I am curious about the technical detail, perhaps, of your work. I wonder for how many days you can measure the thing when you do the 2-photon imaging. How many days you are able to measure the signals in the same animal? I guess that it gets a little bit diluted, or
no, it's a good question. In this case, there are 2 days in between. Structurally, they're quite stable, the axons, so they don't change much, which, I mean, it could be a limitation, of course. But the answer to your question is I don't know. I have not tested this, but that's something that I'm interested in looking at, because we are going to look at plasticity, how it develops over learning. But what I can say is that at least after 2 days, they are very stable, and it's easy to find them again. with the
Okay.
good conditions, with the good technical conditions, it's easy to find them again.
Imagine. Okay, thank you, Belén.
Thanks.
So if there are no questions in the public, perhaps a little bit early today, we can pass to the next speaker.
No questions? Well. The next speaker is Natalia Andersen from INIBIB, Universidad Nacional del Sur. Just go ahead.
Bueno. Well, first, I would like to thank you to the SAN and the organizing committee for accepting me to share my results with you. Today, I'm going to talk about the protective roles of imidazolium salts in C. elegans models of stress and neurodegeneration. Well, we all know what aging is and how our body changes over time. And even aging is not a disease itself. We know that it is a main risk factor for the development of age-related neurodegenerative diseases. For example, see below how fast the prevalence of Alzheimer's and Parkinson's disease increases as we age. All our cells have protein quality control mechanisms to preserve the stability and functionality of our proteomes. This is the so-called proteostasis network. And however, during aging, there is a functional decline of these systems, leading to the accumulation of misfolding proteins, which may aggregate, and this leads to proteotoxicity, and this loss of proteostasis is the main risk factor for the development of age-related neuropathies. The study of this type of age-related disorders is a bit complex in mammals since they are long lifespan. On the other side, C. elegans has a very short lifespan of around 3 weeks, and thus provides an excellent platform for the study of this type of disorders. Besides their short lifespan, these organisms has a very simple nervous system with a purely described neural connectome. And there are also several transgenic models for these proteotoxic diseases. And additionally, these organisms has a huge genetic homology with the human genes, sharing many biochemical pathways. And they are also very easy to genetically manipulate. So we can have mutant and transgenic animals in a very simple way. Well, in this work, we use C. elegans as a platform for imidazole derivative screening. Imidazole compounds are very attractive in the pharmaceutical industry, since by adding substitute, we can generate a wide diversity of molecules with a wide diversity of biological effects. For example, below you can see some synthetic imidazoles in market. In this work, our collaborator, Dr. Silvestri, from the chemical department of our institution, synthesized a series of imidazole compounds, and given the wide spectrum of biological activities of these compounds, we want to explore if these imidazole derivatives have anti-prototoxic roles. To answer that, we perform an oxidative stress resistant assays. The reasons behind that, it does ROS through activating or through acting through proteotoxic mechanisms, lead to proteotoxicity and increase the vulnerability to develop age-related neurodegeneration. So we ask if imidazole derivative compounds could inhibit at some level the ROS proteotoxic action. We perform the oxidative stress resistant assay by synchronizing worms in dishes containing compounds C1 to C5, and at larval for stage, we expose those animals to the oxidant iron sulfate and measure survival. As you can see in the graph, compound 1 and compound 3 increase animal survival to oxidative stress. And then we run some in silico experiments to analyze to predict their pharmacokinetics. And well, for time reasons, I didn't show the results, but we found that compound 3 has a better predicted oral bioavailability. So we continue our studies using this compound. And for now on, we call this compound MSI. So next, we explore the mechanism of action of this compound. One possibility could be that it acts as a direct antioxidant. And so to answer that, we measure the oxidative reduction potential, and we found a positive value, and only negative values are associated with the antioxidant activity. So then we start thinking in a specific mechanism of action. One possibility could be that this compound is activating a dietary restriction program. This is because it is well known in lots of organisms that fasting improves longevity and stress resistance. So to analyze food intake, we measure pharyngeal pumping. Specifically, we measure how many times a worm pumps per minute. And as you can see in the graph, MSI does not modify the pharyngeal pumping rate. So next, another possibility could be that this compound could act by lowering the developmental rate. This is because there is a trade-off between growth and stress resistance. Sometimes organisms that are highly resistant to stressors or develop at a very slow growth rate. So we measure larval developmental rate, and as you can see in the graph, MSI does not modify larval developmental rate. So then we start thinking in the activation of cytoprotective genes. Specifically, we evaluate if MSI acts through the activation of transcription factors that act downstream to the insulin pathway. These are DAP16, KIN1, and HSF1. These transcription factors, when active, translocate to nucleus and activate a cytoprotective transcriptional program, increasing stress resistance, longevity, and improving protein homeostasis. So to analyze the contribution of each of these transcription factors in the MSI mechanism of action, we perform oxidative stress resistant assays, in this case using mutant words for each of these transcription factors. As you can see in the graph, MSI still protects DAP16 and SKIN1 mutant animals, indicating that these transcription factors are not required for MSI mechanism of action. So then we also, however, we observe that MSI protective effects are abolished in HSF1 mutant animals, indicating that this factor is required for MSI protective effects. Then to confirm these genetic studies, we perform microscopic analysis by using a transgenic strain that expresses GFP under the control of the heat-shock protein 16.2. This protein is a downstream effector of HSF1, and as you can see in the graph, MSI increases expression of this chaperone. So we concluded that MSI induces stress resistance is mediated through the HSF1 pathway. Well, so far we have identified that MSI enhances oxidative stress resistance by activating the HSF1 dependent pathway. And we also know that this pathway activates a transcriptional program leading to an improved proteostasis. So next we ask if MSI, through the activation of HSF1 dependent pathway, could be able to reduce the proteotoxicity of age-related proteinopathies. However, C. elegans has no orthologous gene for the proteins implicated in these disorders, like beta-amyloid, alpha-synuclein, or Huntingtin. Anyway, there are several transgenic C. elegans models of these proteotoxic disorders. In this work, we use models for Alzheimer's, Parkinson's, and Huntington's disease. As a model of Alzheimer's disease, we use a transgenic strain that expresses the human amyloid peptide under the control of a muscular promoter. So these animals show an age-dependent protein aggregation in muscle cells, leading to a progressive locomotion impairment that ends in a paralysis phenotype. Here in the videos, you can see how a control worm moves back away from the stimulus after applying stimulus with a fine hair. And on the other side, you can see how paralyzed is a diseased animal of the same age. Well, so we evaluate if MSI has a protective role in this Alzheimer's disease proteotoxic model by measuring paralysis. We measure paralysis both at basal condition and after applying a brief proteotoxic heat shock. In both cases, we observe that MSI delays paralysis decay, and also MSI increases the proportion of moving animals. So we concluded that MSI ameliorates locomotion decline in an animal model of Alzheimer's disease. Then to analyze if this is a disease-specific mechanism or a general one, we also evaluate if MSI has an anti-proteotoxic role in another models of proteotoxic disorders. As a model of Parkinson and Huntington disease, we use transgenic strains expressing alpha-synuclein or 40 glutamines, again, under the control of a muscular promoter. So again, there is an age-dependent protein aggregation in muscular cells with a progressive locomotion impairment. As expected, Parkinson disease animal models show an impaired locomotion, and when these animals are grown under the presence of MSI, there is a delay in the age-related locomotion decline. We observe the same situation in the Huntington disease model, where when these animals are grown under the presence of MSI, again, there is a delay in the locomotion decline. So besides analyzing locomotion, since in these transgenic animals, the transgene is coupled to fluorescent protein, we can also quantify protein aggregation. In Parkinson's disease model, we observe that MSI decreases the expression of alpha-synuclein. So this is in agreement with locomotion results. On the other side, surprisingly, we were not able to find changes in number of aggregates or their size in the presence of MSIs in Huntington animals model. Maybe this could be a consequence of maybe the number of aggregates is not correlated in this model with the toxic effects. So as a conclusion, as a summary, we conclude that MSI enhances animal resistance to oxidative stress. We also found that these protective effects depend on the HSF1 dependent pathway. And also we found that MSI delays the onset of locomotion decline in animals models of proteotoxicity. So through this work, we have identified MSI as a potential therapeutic agent that may delay proteotoxicity in vivo. Now we are trying to explore MSI effects in other organisms like Drosophila. We are also, all the experiments I told here are performed manually. So now we are trying to set up a platform in order to perform a automatized analysis and screening. You can stop by and take a look at Stefano's poster. And also, most of these models are constitutive models where the protein is always expressed in the animal is born. So now we are trying to generate, or we're thinking in generating inducible models in which we can trigger protein expression in adult animals. Well, thank you. And I would like to, these are all the members of my life. I would like to thank to all of them, in particular to Maria Jose, who is leading this line of research with me. Thank you.
Thank you, Natalia, for a nice talk. Questions?
Hi, Natalia.
Hi.
Thank you for your talk. Amazing. I don't remember which were the promoters that you use for synuclein and Huntington models or Alzheimer. Do you know if these promoters are induced by HSF1? I mean, if you are thinking that the mechanism is through HSF1.
Yes, I get it. Good question. I'm not sure about that. I'm not aware of this is happening. I know that these promoters are regularly used by different researchers to study the protein aggregation because they are very simple and as a useful way to study protein aggregation in muscles with as a simple readout for proteotoxicity. I'm not sure about that. Hey.
Hi, Natalia. Thank you for your
Hey,
talk.
thank
So
you.
I was wondering about that, the promoter use. I understand that using a muscle promoter is easier as a readout, right,
Yeah.
for the phenotype. But in terms of relevance for the neuron disease, have you tried to use strains with proteinopathies in neuronal cells and try to read out indirectly the effects of these compounds?
Yes, we are now trying to set up these strains. Of course, these strains that or expressing the proteins under neuronal promoters are a more relevant model, and we use these muscular models as a simple readout. But of course, I agree with you that using neural promoters can provide some additional results. Anyway, there are established models, so we can quantify like aggregation and correlate this with the cell toxicity and measuring locomotion as a proxy of cell toxicity. So yes, I agree with you. We need to explore some neuronal strains, but they are muscular models are very established models for the study of this type of disorders and are very good for performing fast screenings.
Thank you, Natalia. Well, we
Okay.
move on to the next
Thank
speaker.
you.
We will have now the second virtual presentation by Nicolás Martinez. He's in Chile now. He works at the Fundación Ciencia y Vida and University of San Sebastián. And he will present his work, synaptic PKR and EIF3A activation involved in amyloid beta-induced synapse degeneration. Thank you very much. Nicolás, are you there?
Thank you. Do you hear me okay? Do you hear me? I don't hear your answer. Okay. So thank you very much for the invitation. I work, as the presenter said, at Fundación Ciencia y Vida in Santiago de Chile. And we are particularly interested into the regulation of the synaptic proteostasis. So I selected for today's presentation a small history. We are actually starting with related with the regulation of integrity of synapses under amyloid beta induced synaptotoxicity. So I wanted to start with this verse of Alejandra Pizarnik, Argentinian great poetess. In the brain, small flowers dancing like words in the mouth of a mute. These beautiful words emerge of the activity of the entire brain, and to have these small flowers inside our heads, we have to be able to maintain a strong regulation of how the small structures of our brain are interacting with their environment and how they can persist and also change. So there is this very, very common paradigm on neurobiology that for example, the memory function relays on the dynamics that are happening on the lower and most simple levels of the brain. I will not haunt you with the details of this, all these regulatory levels of memory function, but I would like you to have in mind this idea that what happens here at the very small level of synapses, even inside the synapses, is directly related with how the organisms ultimately have their behaviors and neurocognitive functions. So it's pretty important to understand what is happening in this small level. Okay. Sometimes things go very wrong. And for example, at the Alzheimer's disease, it has been described that before the appearance of the symptomatology associated with this disease, for example, the memory loss, we all know, at the very early stages of this disease, actually what happens is synapses lose control of how many proteins they produce and how many proteins they degrade. And protein starts to accumulate around synapses. There are very paradigmatic proteins such as A beta and tau that probably we have heard about in the past. So please keep in mind, again, this idea that the early stages of Alzheimer's disease, there is synapse loss associated with a loss of control of how many proteins are located at this very small part of the brain. Here are some data related with Alzheimer's disease patients, post-mortem electron microscopy, and you can compare how many synapses these patients have. So on the asterisk, you can see the synaptic contacts in a healthy old human patient that has died recently. Oops. And in comparison, the number of synaptic contacts in an early Alzheimer's disease patient, when one can quantify the number of synapses in a certain specific area, for example, in the hippocampus of the patients, one can see there is a strong correlation between a decrease of the number of synapses and the emergency of the symptomatology of Alzheimer's disease. Also, the more synapses you have here in graph C, the more words you are able to remember. Okay, so the integrity of synapses is directly regulated with neurocognitive function, and synapse loss is the strongest correlative of Alzheimer's disease progression. If we go smaller and smaller and on what is happening here, it has been described that on in vitro cultures of hippocampal neurons, that these A beta oligomers that accumulate during Alzheimer's disease are preferentially accumulating around synapses. So here in this immunofluorescence, we can see synapses stained in red and A beta oligomers stained in green, and in these yellow spots are the synapses surrounded by A beta oligomers preferentially. And in the part B, we can see that if we overexpress A beta oligomers precursors and stain it in red, we can see a significant reduction in the number of dendritic spines inside brains. So what happens in the vicinity of synapses and this accumulation of A beta oligomers is toxic for synapses. So we have this local effect of A beta oligomers that is accompanied by a strong toxicity in vitro. Hippocampal neurons suffer of loss of dendritic spines and synaptic contacts in the presence of A beta oligomers. So we have these two proteins that are markers of post and pre-synaptic densities, and they significantly reduce under A beta oligomers presence. Other things that happens in the presence of A beta is significant increase in local protein synthesis, and also the transcription of certain specific proteins. And this was what caught our attention, because actually we are working with a pathway called the integrated stress response. One of the components of this pathway is this kinase PKR, that is a sensor of double-stranded RNAs, which phosphorylate this executor core that is EIF2 alpha, which in turn ultimately leads to control of protein synthesis and the transcription of certain specific proteins such as ATF4. So having this in mind, we have the idea that maybe this pathway was relevant to the integrity of synapses under A beta oligomers neurotoxicity. There are also other data related. I'm gonna go faster to do not haunt you with so many information, but it has also been established in a mutant model that reproduces Alzheimer's disease, the 5XFAD mouse, that if you modulate the activity of this protein, you will more or less reverse the inability of hippocampus to normal firing, okay? So having this in mind, we had this following hypothesis. Local PKR activation participates in A beta induced synaptic toxicity. So very, very focused on the small synaptic environment. And I'm going to show you some results of these two specific aims. We wanted to characterize the localization and functionality of PKR in synapses. It has never been explored before whether this pathway is actually present inside synapses, and also explore which role would this pathway have in the toxicity of A beta oligomers at synapses. So very, very simple question. Where are these proteins located in neurons? So we performed neuronal cultures of hippocampal neurons, and we stained against PKR and the executor core of our pathway, counterstained with a general cytoskeleton marker. And we started to explore whether this protein was present at all neuronal regions. And we actually find that both PKR and the executed core was more or less very well distributed along neuronal cell bodies, as you can see in the somatodendritic compartment and also in axons. A very interesting detail was that when you look closely, you can see that there is a puncta pattern that is characteristic of PKR and phosphorylated form of PKR distribution along neuronal processes. The executor core is also present in neuronal processes, not as discrete as PKR, but also present and concentrated in certain areas. When you perform an histogram of the intensity of this mark, you can see that these proteins are actually present on neuronal processes and are more or less concentrated in certain specific areas that we can see here on those peaks. Also, we have performed three-dimensional reconstruction of neuronal branches that express EGFP protein in green, and we have performed immunofluorescence under these conditions. After the three-dimensional reconstruction, we have found these clusters of PKR and its phosphorylated form inside the dendritic spines at several developmental stages in vitro in those neurons. So this is the first time that this pathway has been located in the vicinity of synapses. More immunofluorescence, this time staining against presynaptic marker VASUN and postsynaptic marker PSD95. And as you can see here, both PKR and its phosphorylated form and also the executed core of our pathway are present in the vicinity of the pre- and postsynaptic events. So, for example, here in this yellow area, you can see more or less that they are very, very close. But to determine whether PKR and the executed core is actually present inside synapses, we have to perform correlation of intensities under confocal microscopy. That one is one of the techniques we use. So actually, you are able to determine whether these the intensity of these signals correlate or not, and this correlation degree gives you an idea of how near are these staining. So you have several possible outputs on this analysis: exclusion, colocalization in a partial way, or full colocalization that will lead you to certain type of graph that I will show you on the next slide. And you can actually also put in testing the randomness of this stain to see whether this correlation is random or is causal through the Van Stielsen analysis. So we perform this, and in these immunofluorescence images, we have performed the intensity correlation analysis. As you can see here in the insets, it's more or less clear that they are very near and correlating, and we have found this pattern of partial correlation between PKR and the pre- and post-synaptic events. And the numbers support this idea that PKR is correlating with the pre- and post-synaptic events, and this relationship is causal and not random. Also, for the executor core, we have performed the same type of analysis, and we have found more or less the same type of results. The executed core of our pathway is also correlating partially with pre- and post-synaptic events in a causal way. Finally, I will show you the last results that we are exploring the presence of these components in vivo in a model of synaptoneurosomal extraction. We lysate brains or hippocamps or certain specific regions we are interested in, and we are able to obtain and extract enrich in synapses, as you can see here in electron microscopy. And when you separate these several fractions, you stain with the same markers, post and pre-synaptic markers. And you can see here that in the synaptic enriched fractions are also enriched in PKR and its phosphorylated form, and the executed core is more and is not such as enriched, but also present in synapses in vivo. Finally, we are performing activation assays in the presence of A beta oligomers that we produce as conditioned media. We add this conditioned media that is enriched in A beta oligomers to our cultures, and then we stain again against the phosphorylated forms, for example, of the executed core. And when we look specifically at the regions where synapses are located, we see a significant increase in the phosphorylation of the executed core. So this pathway is not only present at synapses, but also it's active at synapses under A beta oligomers stimulation. We have also found the components of this pathway in the synapses of 5XFAD mouse that is a model of Alzheimer's disease. So we are at the beginning of a story to explore whether this pathway is able to modulate the toxicity of A beta oligomers in synapses. This is the last result I'm going to show you. And we recently found that when you add these A beta oligomers to our cultures, they have this toxic effect, and there is a significant reduction in the number of synapses. And when you co-treat with an inhibitor of PKR, you more or less have a neuroprotective effect, and the number of synapses are preserved. So thank you very much for your attention. We are reporting for the first time that this pathway is present at synapses, and it seems to have a certain degree of control in the toxicity of A beta oligomers over synaptic integrity. Thank you. I would like to thank to the lab and the head member of the lab, Dr. Soledad Matus, and all the lab members and collaborators. Thank you.
Thank you very much, Nicolás. Is there any question in the audience? Not. No people working on that. Nicolás, from your
Yes.
result, I wonder if you think that the PKR pathway could be more usefully targeted at the early stages of this pathological condition, or you are thinking more about the advanced stages of the Alzheimer's disease?
You mean the activation of the presence or the presence?
The activation, yes, of the pathway.
That's an interesting question. We actually have not explored the dynamics of activation yet, but we presume that PKR is activation is an early marker of the synaptotoxicity of A beta oligomers. Because, for example, there are some reports on patients that show that phosphorylation of PKR is increased before the symptomatology, but specifically what happens inside synapses has not been explored. So that's probably one of our next steps.
Okay, thank you. So
Thank you.
if there are no questions, I don't know, we can pass to the following speaker. Thank you, Nicolás.
Thank you.
Thank you.
Our next speaker is Mariela Trinchero from Neuronal Plasticity Lab from Fundación Instituto Leluar.
Hi, everyone. I want to thank the SUN organizers for giving me the opportunity to present my work today. I'm very excited. And yeah, the title of the talk is Activity-Dependent Plasticity of Neurons Born in the Aging Hippocampus. So you might have probably heard that the generation of new neurons in the adult brain is a conserved process across all these species, and it has been actually reported in humans. But of course, in the lab, we focus on mouse models to study adult neurogenesis. And in mice, there are two restricted zones where adult neurogenesis takes place. One of them is the subventricular zone, and the other one is the subgranular zone in the hippocampus. So in particular, in the dentate gyrus, there are these neural stem cells that give rise to granule cells, which are the principal cells of the dentate gyrus, and they will, with time, develop and integrate into the pre-existing circuit. So, slow, but okay. So the way in which we study adult-born neurons is by tagging them either with a retrovirus, which will only label dividing cells, so that allows us to follow the neuronal maturation of these neurons, or this really cool tool, which is the ASCL1 tomato mice, and that allows us to do the same thing with the tomato reporter. So with these tools, we are able to perform electrophysiological recordings of these cells and confocal microscopy. So here is a really beautiful picture taken by Diego in the lab, showing how you can visualize adult neurogenesis in the ASCL1 tomato mice I showed you before. So you can see here a label in red all the new granule cells that were born during adulthood. So basically, these cells project their dendrites towards the molecular layer, where these are the input cables, and through these cables, they receive somatosensory information coming from the entorhinal cortex. So they receive these informations here in the molecular layer through the dendrites, and then they project their axons through the hilus towards CA3 in order to connect with pyramidal cells and GABAergic interneurons. So we were interested a long time ago, when I was young, to study the effects of aging in adult-born neurons. Why? Because we did know that during aging, the number of neurons that are being generated decreased sharply, right? But the thing that we didn't know is what is the quality of these few neurons that are being born. So to study this, we injected the retrovirus expressing GFP in the dentate gyrus in either young animals, meaning 2-month-old, or 8-month-old animals, which are kind of middle-aged, kind of like me now. So we waited 21 days to sacrifice the animal and perform immunofluorescence and confocal analysis. And as you can see here, there are two main things that you can say. First is, I'm too short, is that the number of neurons, of course, was decreased, because you can see here that the young animals have a lot of neurons and this one doesn't. But also you can see that the dendritic tree is not well developed, it's really short, also like me. And it doesn't even reach the molecular layer where, sorry, because where it is supposed to receive afferents coming from the entorhinal cortex. So we can assume, and we know because we perform electrophysiological recordings of these neurons, that they are not connected into the circuit. So we were wondering what could be supporting this slow maturation. And we know that for the hippocampus, I mean for all the brain, but especially in the hippocampus, activity is really important and can highly impact neuronal development. So we decided to assess this by studying the density of ARC, which you probably know that is an immediately gene. So if you see a lot of ARC, it would mean that the granule cell, in this case, is active or more active than if you see low density of ARC. So as you can see here, the density of ARC is really decreased compared to young animals. So aging delays morphological maturation, and we saw that this happens especially during the first weeks, but then the neuron gets to develop to final stages. And this is supported by local activity and diminished levels of neurotrophic factors that, because of time issues, I cannot show you, but we measure. So far, aging doesn't look that much fun, right? But there is hope, believe me. So I told you that activity is really important and can shape neuronal development. And we know that there are hippocampal activity modulators that can change the pace of the maturation of these neurons. So we know that one of them is voluntary exercise. So first we decided to study the effects of running on these neurons. So for this, we did a similar experiment. We injected a retrovirus expressing GFP in 8-month-old animals, waited 21 days, and one set of animals was exposed to a running wheel. So in the control group, you can find these immature-looking neurons that are not connected. And again, we did the electrophysiology, and they cannot fire action potentials, and they are not receiving glutamatergic inputs. But then, and I have to tell you that these animals run a lot, like 5 or 8 kilometers a day, so that's a lot. And we found this surprising effect of running, in which you can see a significant increase in the dendritic tree length. And just as a reminder, here it's a mature neuron born in a young mouse. So you might think that these look really similar. I mean, more similar than this, of course. And it's kind of like the running animals rejuvenated their neurons. So I bet you are all going for a run tomorrow. So I show you that running can accelerate neuronal maturation in the aging animals. And another stimulus that we know that can modulate neuronal maturation is an enriched environment. So what is enriched environment? And let's hope this video works. So this is an enriched environment. This is a larger cage that the animals are used to. And yeah, they are exposed to toys and tunnels where they can explore, and they even have a good house where they live rent-free, so they are really happy. And yeah, so this is really a strong stimulus. So for this, we did the same experiment than before, but exposing animals to enriched environment, and we saw again a positive modulation of neuronal development of the dendrites in these animals. And we also perform another other experiments, but for time issues, I cannot show you. You can check the literature. So I'll show you what, okay, something needs to happen. So far, I have shown you that these two stimuli can modulate neuronal development. And nowadays, we are interested in understanding how gamma stimulation can modulate neuronal development. And I tell you in a minute why. I've been talking to you a lot about activity and how important it is in the hippocampus. And you probably also know that oscillations are the features in the brain that enable synchronization of neural activity between brain regions, right? So it enables the different areas of the brain to communicate with each other through activity. In particular, gamma oscillation, which goes from 30 to 100 hertz, or this can vary depending on the literature, but has been really involved in cognitive functions such as attention, navigation, and sensory processing. Moreover, several labs have shown that in Alzheimer's disease models and aging mice, the power of gamma oscillations is decreased. And this correlates with an increase in A beta plaque formation and impair memory. So the lab of Li Weizai in MIT have been studying how to restore this gamma power. And for that, she decided to flash LED light at 40 Hertz, which from now on I will call flicker. So she saw that by flashing animals with these lights, they can increase gamma power, as seen here. And also there is a reduced beta plaque formation levels. And also there is a restoration in spatial learning, as you can see here in this water maze experiment. But even though this has been published largely in high impact papers and also on more maybe creepy, there are studies being conducted in humans, but the mechanisms are still not known, right? We don't know what the flashes of light are doing in the brain to get to this. So the remaining questions that arise from this work are what are the changes exerted by gamma stimulation on neuronal plasticity? What are the mechanisms underlying gamma stimulation effects? And in our particular interest, does gamma entrainment affect neurogenesis in the aging hippocampus? So we came up with the hypothesis that gamma stimulation triggers activity-dependent modifications that induce plastic changes in new neurons in the aging hippocampus. So to test this hypothesis, with the help of Emilio Kropp in the institute, we developed a setup of stimulation of 4 hours a night of stimulation at 40 Hertz that are spaced by 2.5 hours of rest. And how it looks the deadlight, like a dancing club, but it's not. And yeah, so basically we did a similar experiment, like a screening experiment, because we know that by waiting 21 days and exposing animals to enriched environment, running, etc., you can increase development. So we try with gamma stimulation. And what we found is that with 40 Hertz, but not 8 Hertz, you can increase twice the dendritic tree length. So it appears that this stimulation, which is non-invasive, can actually do something for these neurons. But until now, I've been talking to you a lot about the inputs of these cells, which are the dendrites, but we are also interested in addressing what are the changes that can happen in the output. So going back to this beautiful picture, I told you that the axon, which is the output cable, goes through the hilus towards CA3. And here, if you focus, you can see the mossy fiber buttons that where you can perform some kind of measurements. So if you measure the area of these buttons, you can have an idea of how connected this neuron is to CA3 pyramidal cells. On the other hand, these mossy fiber buttons also display some extensions that they are connecting with GABAergic interneurons. So the more philopodia you have, the more connected it should be to the GABAergic circuit. So in this same experiment, we studied mossy fiber buttons, and we found that the area of these buttons increased twice. And also we found that while in control animals, the buttons had almost no extensions, in the gamma-trained animals, we had at least one. So I show you that gamma stimulation significantly increased the dendritic tree length, which tells us about the input of these neurons, and also promoted a faster connectivity with outputs in CA3. But this is, of course, not over. And to summarize, I've been showing you that all these stimuli can accelerate neuronal maturation. And the conclusions we can take out is that first, in the aging hippocampus were measured changes are not expected, basal activity is low, and developing neurons appear immature. Activity boosters, such as running and rich environment gamma stimulation, can trigger high levels of plasticity that exceed even those that we have seen in the young animals previously in our lab. Also, light flickering at 40 Hz awakes mechanisms that promote neuronal plasticity, not only in pathological conditions, as was seen by the side group with Alzheimer's disease, but also in the healthy aging brain. But because this is an ongoing project which is growing with time, we decided to explore more questions. How does gamma flicker affect the electrophysiological properties of the new GCs? And we are performing these experiments right now, not right now, but you know. And then do neural progenitor cells benefit from gamma stimulation in aging animals? And this is important because of this decrease that I show you of adult neurogenesis, right? But maybe gamma stimulation can increase the number of neurons that are being generated. And we are lucky enough that we have a lot of talented people around, so we can collaborate and answer a lot of questions with different techniques. We are wondering whether a gamma stimulation can improve spatial learning in aging mice, and if these effects are mediated by adult neurogenesis. And for that, we are collaborating with Nati and Vero in the lab, who are performing beautiful behavioral experiments. Also, we are interested in studying whether flicker exert network remodeling in the hippocampus. And to answer this question, we are working with Mati and Emilio, which can perform these really cool in vivo recordings and optogenetics. And last but not least, we are interested in studying the molecular mechanisms that are underlying this positive modulation. And for that, we are collaborating with Paola Arlota in Harvard to perform RNA-seq analysis of these neurons that are being modulated, and with Ariel Chernomorets, who is a magician with the bioinformatics. So I want to thank this beautiful team that I'm so lucky to work with them. This is Ale, our group leader, and I want to especially thank Maggie, which has been my amazing friend and partner in crime in all this aging adventure, and also Agos, which has joined us recently with the gamma stimulation project. And I invite you to go to their posters to learn more about transcriptomics in aging hippocampus and gamma stimulation. And just as a publicity, we have an open postdoc position. So if you like this project and you want to talk to me or write an email to me or to Ale, we are looking for people, and you can follow us on Instagram. Thank you.
Thank you, Mariela, great talk. I have a question. There's any specificity with the gamma flicker, or have you tried 1000 hertz or 10 hertz, or if it was the sound? Because for the video you showed, it was kind of noisy.
Yes.
It was,
Well,
I was thinking it was just enrichment, or there's any specificity.
Sure, that's a great question. Well, first, I have to say that this investigator site had shown that with other frequencies, there is no effect of the stimulation. And then in our hands, we try 8 Hertz, and we saw that nothing happens. And then we also try, and I'm spoiling Agos' poster, but we try only only light, and then light plus sound at 40 Hertz. And we show that the increase in the neuronal maturation is the same. So there is not like a synergistic thing with audio and visual stimulation.
Mariela, I have 2 questions. One is, light is usually not something that mice like.
So this looks more like an aversive signal for
Mhm.
the animals. Do you agree, or is it that the flicker is not perceived for the animal as a source of light that they would
Yes.
escape from?
That's a good question, and that, yeah, that was a concern because when we first, I mean, this line is new, so when we first saw the flashes, we were like, okay, this is not nice. But it has been shown that actually these lights have an anxiolytic effect on animals. So it might be stressful for me, but not for them. So yeah, that has been addressed, and there is no stress coming from the lights.
And the other question is, in our world, we think more is better. And so growing more dendrites seems like, oh, they are going to do better. Is that right? Is it always the case that more dendrites is better?
Well, I think that in the condition of aging, where you can see some cognitive decline, having dendrites more connected might be beneficial. It's different from some kind of aversive stimuli where, for instance, in epilepsy, you have a dendritic growth, but this is aberrant and it generates hyperexcitability, and it's not good. But I think, yeah, I think you should go for a run.
So the last question is telling jokes a condition to show in the Schindler lab, or?
Sorry about that, I'm nervous.
Questions? Anyone? No? Well, we go, thank you, Mariela. We'll go to the last speaker.
So we are good in time. So we have our last speaker, sorry, who is Leonardo Versaci from the Universidad Nacional de Quilmes. And he will present us some nice results. So thank you very much.
Okay.
Leonardo.
Thank you to the organizer for the invitation. I'm going to show a main part of my PhD, which I finished last year. So is it ready, the presentation? Okay. I worked in LDSM lab. Held, it's not that. It has the word synchronization in his title. Okay. It's coming. Okay. Okay, we're going to talk today about time processing. This is an important function of our brain because it's related with daily behavior. We perform time processing in different time scales, for example, in circadian timing and microsecond timing. The mechanism behind these scales are relatively well known, but for the other two scales, interval timing and millisecond timing, the mechanism behind are not fully understood because maybe it's more complex. We are going to concentrate today in these scales, normally related with motor control, to speech, to performing a musical instrument, and with this behavior, sensory motor synchronization that we are going to study today. So let's define sensory motor synchronization. This is an almost human skill, consists in synchronizing motor actions with external periodic stimuli, as when we dance or when we tap our finger to the music. It requires temporal prediction in a sense that subject must predict when next stimulus is going to occur to match motor action with it. So the motor action begins before the presentation of the stimulus. This is in contrast to what happened in a reaction time task in which the action begins after the presentation of the stimulus. And despite this apparent simplicity, it depends on a lot of factors such as tempo. This is the period of the sequence. The stimuli modality can be auditory, visual. The factor employed, it can be the foot, can be the finger, and perhaps it depends on this factor. Attention barely studied in the past. So based in preliminary results, we decide to study the effect over the performance, and we pick up a paradigm called paced finger tapping to study. This is a widely used task because it allows us to study high function of the brain. It has a lot of variants. We are going to use an auditory stimuli. We call them beeps. This is like a metronome. Also, we are going to use a second beep. This is an additional auditory feedback that subject here every time they touch the sensor, and this allows us to manipulate the sensor information that subjects are receiving. And also we are going to use tempo perturbation. This is a sudden and unexpected change in the period of the sequence that enable us to study the resynchronization process after a perturbation. Okay, few words from attention. Really, research in attention is a very active field, but attention can be oriented to a specific stimuli or to a specific modality. In this case, you can orient attention to time aspect. In reaction time paradigm, orienting attention in time has been studied by Nover and collaborators, and they conclude when attention is directed to a specific point in time, performance is improved or reaction time decreases. In paced finger tapping, attention has been studied a few times. In this work from Miyake et al. In 2004, they conclude that finger tapping is an automatic behavior, not depending on attention. We are going to discuss this affirmation. They use a dual task paradigm. This is a paradigm in when you divert attention from the primary task to a secondary task. We are going to propose also a new method to manipulate attention without using a dual task paradigm. Okay, let's define the main variable that emerged from the task. It's called asynchrony. These vertical lines represent the stimuli, the beeps. We start with a period of 600 milliseconds. This is the perturbation. You hear something like beep, 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 and you have to synchronize finger movements with this stimuli. In a similar way, we can represent responses with these vertical lines. So we define asynchrony as the difference between responses and stimuli in a way that if asynchrony is 0, it means that responses and stimuli occur at the same time. And if asynchrony is negative, such as this example, it means that responses occur before a stimuli. It's important to note that perturbation is unexpected, so this is really a forced error, and normally subject resynchronize in a few beeps. Okay, this is the experimental design. We propose to answer this question, if attention improves performance. We use a full factorial design with 3 factors. One of them is attention with 2 levels, normal attention, high attention. The other factor is the additional auditory feedback with 2 levels, without feedback and with feedback. And the third factor is perturbation sign with positive and negative levels. So defined in total 8 conditions. As we want to propose a new method to manipulate attention, we wanted to be sure that subjects were really tapping in 2 different levels of attention. So we measure 2 correlates of attention. One of them is the auditory earpiece. We employed an emotive EEG with 14 channels. This is a portable EEG. And the second correlates, it's a variable called workload. This is a subjective report of the mental effort employed in the task. This is the way how we manipulate the attention. We use two sensors, one next to the other. In normal attention, we really didn't give subject any specific instruction about where to orient their attention. The sensor in the left is not used in this condition, and this is the most common instruction in all the experiments of finger tapping. But in high attention, we ask the subject to start tapping in the sensor on the right, and when they hear the tempo change, the perturbation, jump immediately to the other sensor and continue tapping there. In this sense, we say that attention is oriented to the temporal aspect because subject must be aware of the tempo change. So we measured 44 musicians, and to avoid carryover effects, we divided them into groups, 22 for normal and 22 for high attention. So this is real data for one subject in one condition, a temporal series of asynchrony. We're showing here mean asynchrony. And in the horizontal axis, the bip number relative to the perturbation. The perturbation is represented by this vertical line. We see here the resynchronization process. It's important to know that one trial for us is not just one bip. We are seeing here 22 bips, and we are averaging here 6 trials of the same condition. We can see that mean asynchrony in this region, pre-perturbation, is negative. Remember that it means that responses occur before the stimuli. So perhaps this is the most replicated phenomenon in finger tapping, and till today there is no single explanation, or not a complete explanation, because it depends on a lot of factors. Okay, let's see some of the main results regarding to mean asynchrony in pre-perturbation region. We wanted to see, we see that, okay, now we see 4 conditions because perturbation sign factor here is average. So the first 2 columns represent normal attention without and with feedback, and the second 2 columns represent high attention with no and with feedback. We see that mean asynchrony decreases in absolute value depending on both factors, attention and feedback, but only attention has a significant effect over the mean asynchrony. Regarding the standard deviation of the asynchronies also in pre-perturbation region, we see that this variable depends on both factors, but in this case, none of them has a significant effect. So, taking into account these 2 results, we can say that finger tapping is affected by attention, at least in its stationary phase or pre-perturbation phase, because subjects are more accurate in high attention, or performance, we can say, is improved. The results regarding the attention correlates, we are seeing here 2 ERPs, one for normal, one for high, aligned to the stimuli. We found a significant difference in this shadow region. And it is known from literature that this kind of difference in early components of DRP is related to difference in attention. And specifically, the ERP with lower amplitude in P2 is related to high attention. The result for workload, the subject in high attention report more mental effort. So this is related with attention too. So again, taking into account these 2 results, we can say with some degree of confidence that subjects were really tapping in 2 different levels of attention. Remember that we want to propose a new method to manipulate attention. And the result regarding the resynchronization process, this region in green here, we use an index called resynchronization efficiency that takes values between 0 and 1. And now we are seeing here 8 conditions because perturbation sign is unfold, but there is a lot of information. I want to concentrate in the attention effect. We see a significant effect of attention over the resynchronization efficiency. We can say that subject in high attention, these 4 bars here, are more efficient to resynchronize than subject in normal conditions. So again, we can say that finger tapping is affected by attention also in the resynchronization process. So we can summarize these results. We saw that attention improves accuracy. This refers to the stationary phase. Subjects were more accurate in high attention. We recently saw that attention increases resynchronization efficiency. Subjects were more efficient to resynchronize after a tempo perturbation. So we can say in general terms, looking this both result, that sensory motor synchronization is affected by attention. And this is in clear contrast to what Miyake et al., the work I shown recently, said about attention. Remember, they said that finger tapping was an automatic task, not depending on attention. And maybe because they used a dual task paradigm. So now I'm going to say a few words about it. But from a methodological point of view, perhaps it's better to start controlling attention because it has an effect over the performance. And the second important conclusion is attention can be manipulated without using a dual task paradigm. This is important because a dual task paradigm is common in our field, but I think that dual task paradigm is useful when you want to see if 2 tasks are sharing some resources because you're going to see some interaction between them. But if you want to, if you only want to have different levels of attention, such as low attention, high or normal, it's better to use a paradigm without dual task, sometimes very complex. Okay, these results were published recently with Rodrigo Laje in a publication from the last year. And that's all. I want to thank my compañeros here in Quilmes, and thank you.
Thank you very much, Leonardo. You have a question for, I think that everybody's wanted to go to.
Okay,
take
me
lunch.
too.
But I wonder why you only use musicians for your studies.
Okay, yes,
But I guess
because it is known that musicians have a lower variability in their performance. So if you want to see a small effect, it's better to work with musicians. We tried with non-musicians, but they couldn't finish the experiment.
okay, it's good answer. So, well, thank you very much,
Okay.
Leonardo. If we don't have more questions, we can go to have lunch. And then at 2:30, we have the first poster session at the IFIBINE. So thank you very much for being here.